Hello, welcome to a new video class. In this video class, we are going to study mainly the topic purification and refolding of proteins. Especially, we are going to deal with recombinant protein. We already studied in biotechnology about heterologous gene expression, that is expression of a particular gene within a foreign cell. So during the production of proteins from an heterologous gene, that protein may not be of a functional one. So we need to purify that protein by refolding in different ways. So here we are going to deal that purification and refolding process. So before going to the refolding and purification of recombinant protein, that is the protein produced by heterologous gene, we need to study few terms so that we can easily able to understand the concept. So now we need to know what is inclusion bodies. Inclusion bodies are the reserve food material present in the cytoplasm of mostly prokaryotic organisms. Or we can say that it is a type of proteinaceous reserve food material produced by mostly prokaryotic organisms. That non-functional, normally that proteinaceous reserve food material found within the cytoplasm of a prokaryotic cells are normally they are non-functional proteins. So such kind of inclusion bodies formation also takes place in case of heterologous gene product or heterologous protein, that is recombinant protein. So when we produce a protein by using heterologous gene, that protein may not be functional. So such kind of protein exists as inclusion bodies within the mostly they are exist within the cytoplasmic space of that prokaryotic organism. So before going to make it economically valuable, we need to convert that non-functional inclusion bodies into a functional normal active protein. So for that purpose we are focusing on purification and refolding of proteins. That's about the concept. So we need to know one more thing about the inclusion bodies. What are the factors that leads to the formation of more amount of inclusion bodies? How inclusion bodies formation takes place inside the cell? So there are several factors that affect the formation of inclusion bodies or that affect the rate of formation of inclusion bodies. Let us try to understand what are all those factors that lead to the formation of inclusion bodies or non-functional proteins from heterologous gene. One is overexpression of heterologous gene. We know that we only select a specific gene or heterologous gene, and that heterologous gene transfer into a suitable host cell to express. So after the transfer of a heterologous gene into the host cell, that heterologous gene can sometimes that express within high rate. So that gene produces more amount of proteins than when it is present in its native cell. So after the transfer of a particular gene or heterologous gene into a host cell, that particular gene that shows an overexpression when compared to the expression of that gene within the native cell. So due to the overexpression of protein, the inclusion bodies formation occurs. So that is the one reason. And second one, if the newly produced recombinant protein or heterologous G product contain more amount of sulfur containing amino acid. Let us take an example, cysteine. We know that cysteine amino acid. Cysteine. That amino acid contains acid COH group, amino group and hydrogen group, and a variable group containing CH2SH, sulfur containing amino acid. If the protein product contain more amount of sulfur containing amino acid, especially cysteine, that sulfur can able to make disulfide bond, creating complicated structure. So that the amount of if the amount of sulfur containing amino acid is high, then there will be more chance for the formation of inclusion bodies. That is the second point. And when we move to the third point, expression environment of protein also play a crucial role in the formation of inclusion bodies. So the expression environment also plays a crucial role. And third one is the lack of post-translational modification of protein. We know that after protein synthesis, the newly synthesized protein may not be functional. That protein needs to modify by using different kinds of enzymes. Such type of enzymes are called post-translational modification enzyme. So that leads to the formation of covalent bonding, phosphorylation, glycosylation, something like that. So lack of such kind of post-translational modification enzymes also enhances the rate of inclusion bodies formation. So these are all the few factors that enhances the inclusion bodies formation in case of recombinant proteins. Now let us move into the purification and refolding methodologies. So there are different kinds of purification and refolding method employed by humans from past. So among these, one is called dialysis. We know that mostly denaturating agents or denaturant. For example, when we take the case of urea is the best example for denaturant. NH urea, we know that the molecular formula of urea, NH2CONH2, it acts as a best denaturant. It can able to prevent the formation of functional proteins. So denaturant. So for example, when we case the case of denaturant like urea, that means denaturant can able to alter the structure of protein. Thereby, it can able to prevent the formation of functional protein that leads to the formation of inclusion bodies. So, in case of dialysis method, we are decreasing the concentration of denaturant in a protein sample. So when the concentration of denaturant get decreases in a sequential way, if we can able to decrease the concentration of denaturant in a protein solution in a stepwise manner, then its action also decreases. That leads to the inhibitory action of denaturant. Ultimately, we can able to reduce the rate of inclusion body formation. Thereby, we can refold the protein in a proper way. Like that, we can also employ dilution method. And like that, we can also employ ultrafiltration method. Among these three methods are usually is an ancient method or traditional method. Even though we are using dialysis, dilution and ultrafiltration method in past, its refolding rate or recovery rate is very, very low. That is why now this kind of dialysis, dilution and ultrafiltration methods are not employed by scientific field, because when we take the economic value, or when we try to produce a functional protein, we cannot able to depend this method because its recovery rate is very low. That is, it is not an economically valuable method. That is why it is not relevant in present condition. Okay. Whereas when we take the fourth case, gel filtration chromatography, it is a special type of chromatography. We know that chromatography is the best separation technique. So by using this separation technique, we can able to eliminate the denaturant and denaturating agents. And we also know that chromatography is a special separation technique. During that, the basic principle of the chromatography is that contains a mobile phase and a stationary phase, and that stationary phase acts as a best agent for removing denaturant from that protein. That means denaturant are mainly responsible for the formation of inclusion bodies. Like that, we can also use another type of chromatography, that is called liquid chromatography. These are the second type of chromatographic technique. And when we move into the third, you can see HIC, that is hydrophobic interaction chromatography. And the seventh one is called IEC, ion exchange chromatography. And eighth one is called gel exclusion chromatography. We can say that GEC, gel exclusion chromatography. And the last one, affinity chromatography. These are the few techniques which are used for the purification and the refolding of protein. Thereby, we can decrease the amount of inclusion bodies or non-functional clumps or clusters of protein. So by this technique, we can refold the non-functional recombinant protein into a proper active protein by a correct folding method. And we also know that proteins formed, the newly formed proteins are called the primary protein. That primary protein is folded into secondary, tertiary and quaternary structure. If they are folded in an improper way, that leads to the non-functional protein or hazardous protein. So in order to avoid such activities, we are depending this method. Okay. Next, we need to know how we can confirm the folded protein be a functional one. That is, how we can able to validate the process of refolding. So it can be done by using different methods. Among one is called electrophoresis. We know that electrophoresis is the migration of charged particle towards the opposite electrode. Suppose this is a positive electrode, this is a negative electrode. You can connect it into a circuit. By using this electrode, we can able to migrate charged particles like proteins by using a medium having definite pore size. Okay, so electrophoresis method can be employed to validate the folded protein, to check the folded protein is correct or not. And like that, we can use another technique called ELISA, that is enzyme linked immunosorbent assay, ELISA. By using this ELISA, the basic principle of ELISA is that it works based on antigen-antibody interaction. By using this ELISA technique, we can validate the folded protein by giving an appropriate antigen or antibodies. And Like that, we can depend blotting technique, mainly Western blotting technique. We can depend Western blotting technique. We already studied what is Northern blotting, Southern blotting, Western blotting. Among these, that Western blotting technique is used for validating the refolded protein. Like that, we can also depend HPLC. Liquid chromatography, high performance liquid chromatography, HPLC. We can depend HPLC technique. It's a chromatographic technique. So we can validate the refolded protein by using different techniques like electrophoresis, by using migrating the particle protein by using electric field, like the ELISA enzyme linked immunosorbent assay by reacting antigen antibody interaction by giving antigen antibody interaction, and like that Western blotting we already discussed, and like that HPLC. Okay, that's all.